Howdy, and welcome to the podcast for Microbiology 205. This podcast will talk about eukaryotic non-membranous organelles and membranous organelles. So remember, the organelles live inside the cytoplasm of the eukaryotic cell. And in this first cartoon, on the right-hand side, the top right-hand side, you can see a sort of a big purple, looks like some kind of catcher's mitt. That is the ribosome, and the ribosome is where proteins are made. So that thing that looks kind of like a comb moving through the ribosome, that's the messenger RNA, and the messenger RNA is being translated using the ribosome and the transfer RNA, which is it kind of looks like a worm sitting in the catcher's mitt. So that worm thing, that's the transfer RNA that brings in those balls, those pink balls, which are amino acids, and those amino acids correspond to the triplet codon on the messenger RNA. And when you link those amino acids together, you make a protein. So that's what's going on in this cartoon. Now, the difference between ribosomes in prokaryotes and eukaryotes is their weight. So the eukaryotic ribosome has a weight of 80 Svedberg units, that's what the S stands for, and prokaryotic ribosomes have a sedimentation rate, which is corresponds to their weight of 70 S. So this is important to know because, oh, and by the way, remember that big purple blob catcher's mitt thing in the cartoon is supposed to be the ribosome. So remember, it's important that you know the difference between the eukaryotic and the prokaryotic ribosomes in terms of its sedimentation rate, because this is where pharmacologists can design drugs to target the prokaryotic ribosome, and then the prokaryotes, like bacteria, won't be able to make proteins and then they'll die. But that same drug won't affect our own 80S eukaryotic ribosomes. And then in the bottom cartoon, it shows a picture of microtubules, microfilaments, and intermediate filaments. So the microtubules, actin, and intermediate filaments, those are the proteins, the molecules that comprise the cytoskeleton. These proteins enable the cell to contract and move around, and also during mitosis and meiosis, this enables the cell to pinch off and form two new cells. So I want you to read through this, turn off the podcast, read through it, and see if you can get the right answer, and then I'll tell you in class. So in this next slide, we'll continue with the eukaryotic non-membranous organelles. This is a transmission electron micrograph, or a TEM, of centrioles that are living within the centrosome. So the centrosome is the space, and the centrioles are the actual structure, the protein structure that lives there. So animal cells and fungal cells, which are eukaryotic cells, they have two centrioles in that region. And then in letter B, it shows that the centrioles are made of these proteins called microtubules. And you use the centrioles in mitosis and cytokinesis. So cytokinesis is just when the two cells that have undergone mitosis are pinching off. So now we'll turn to eukaryotic membranous organelles. So these have a plasma membrane around the organelles, where the previous two organelles that we looked at did not have a plasma membrane surrounding them. So in this cartoon, it shows half of the cartoon is a transmission electron micrograph, and then the other half is a cartoon that sort of continues the TEM. So this is a chloroplast, and it shows things inside the chloroplast that we don't want to talk about now, but we'll talk about that later. But it's just the light harvesting structure. the thylakoid and the granum, that's just the structures that harvest the light waves that come into the chloroplast, and then those light waves are used to make carbohydrates for the plant to eat, and also as a byproduct of that you get you get oxygen. So any photosynthetic eukaryote like a plant or an algae would have these structures, and they would be making oxygen for most of the earth. There's a dual plasma membrane structure, and these structures, the chloroplast, they have their own set of DNA. It's circular DNA, and they also have 70S ribosomes, and that's why scientists came up with the endosymbiotic theory that says that a small aerobic prokaryote found itself inside a large anaerobic prokaryote millions of years ago, and the two cells formed a symbiotic relationship, and we have eukaryote cells from that today. Here's another eukaryotic membranous organelle. It's called the nucleus, and if you look at the cartoon, you can see that there's a dual layered nuclear envelope. Inside the nucleus is chromatin. That's just the DNA in non-condensed form. And in the very center, there's a nucleolus, a structure called a nucleolus, and that's where all the RNA is made for the ribosome, like ribosomal RNA. There's nuclear pores embedded in the nuclear envelope, so things can get shipped out of the nucleus into the cytoplasm. And then if you look carefully at the cartoon, where the nuclear envelope comes out, it forms, where the outer membrane of the nuclear envelope comes out, it forms the endoplasmic reticulum. And then you can see the endoplasmic reticulum is studded with ribosomes, which we talked about a couple slides ago, and that's where proteins are made. So, like I said, the outer membrane of the nucleus extended outward to form the endoplasmic reticulum. In this cartoon, they show transmission. Part of the cartoon is a transmission electron micrograph, and then the rest of the cartoon is an artist's conception, or the continuation of the TEM, and it just shows that it's composed of many membrane-bound, sort of flattened sacs. So once the protein is made on the ribosome, so those little dots on the ER are the ribosomes, once a protein is made, that protein can be shuttled into the endoplasmic reticulum. And then in the endoplasmic reticulum, you can continue making the protein into a mature protein, maybe by putting lipids on it or carbohydrates on it. Once you've got your protein made in the ribosome and the ER and you're ready to ship it out, then you want to ship it to the Golgi apparatus. So again, you can see in this cartoon there's a transmission electron micrograph, and then the rest is the cartoon. It shows in the bottom of the cartoon there are vesicles, so those are just membrane-bound vesicles arriving from the ER with proteins in them. The vesicles fuse to the membrane-bound sacs of the Golgi apparatus, and then they get transported towards the plasma membrane, where ultimately the proteins are shipped out of the cell via exocytosis in secretory vesicles. So then this is just sort of the rest of the major organelles, the lysosomes, peroxisomes, vacuoles, and vesicles. So if you look at the top TEM, this micrograph shows the cell wall. So if you see there's a cell wall, then you know that is most likely a eukaryotic cell that is a plant cell, because plants have cell walls and animal cells don't. Shows the nucleus, and then there's a central vacuole there, and things can be stored inside there, inside the vacuole. Things like starch, so large carbohydrates for food source, lipids, and sometimes it could be a water vacuole. The lysosomes are also membranous organelles in eukaryotic cells. The lysosomes often contain catabolic enzymes, and so those are things that can break down your old organelles, and also lysosomes have enzymes in them that can break down and kill bacteria. And then at the bottom of the screen there's a cartoon of a peroxisome, and we'll talk a lot about peroxisomes later when we talk about metabolism, because peroxisomes contain oxidase and catalase, and you need those if you're a eukaryotic cell and you are using oxygen to do respiration. You need oxidase and catalase because oxygen can be toxic, and you need these enzymes to control the toxicity of oxygen. Another eukaryotic membranous organelle, of course, is the mitochondria. So if you look at the bottom of the page, it shows, again, there's a dual membrane system. There's an outer membrane of the mitochondria and an inner membrane. And where the inner membrane folds, those folds are called crista. And then inside the crista, or the inner foldings and the inner membrane, is the mitochondrial matrix. And remember, the mitochondria is the powerhouse of the cell, so we'll talk about this more in Chapter 5 to see how ATP is made in the mitochondria. And again, this refers back to the endosymbiotic theory for eukaryotic cells, where billions of years ago, or hundreds of millions of years ago, mitochondria were thought to be prokaryotic, small prokaryotic cells that did aerobic respiration that got trapped in a big prokaryotic cell that did anaerobic respiration. So I'll let you read this, and we'll talk about the answer next time we're in class.